Welcome student in the 6th week of the SWAM course Anatomy of Flowering Plant, and in this week we will be discussing about secondary growth. Before this week, whatever we have discussed was about primary growth of the plant, and in which all the structures which were formed were the primary structures. That is, they were mostly responsible to increase the length of the plant. However, plant also increases in width. So in this we will try to understand how this secondary growth takes place and what are the important components which help in this secondary growth formation. Now, the topics which will be covered in this entire week will describe about vascular cambium, because this is the most important structure which is responsible for secondary growth, and how the secondary growth takes place in the dicot root and the dicot stem, because we find that mostly these dicot root and stem has very elaborate or stem or root with very big diameters, or they have very broad stem and root. Then we will try to understand the wood, the composition of wood, see how when the secondary growth occurs, you would have seen that the stem is very broad, and we generally call those part of the stem which are very hard and thick as wood. Okay, so we will try to understand what is actually this wood is, and what is the composition, how different wood structures varies to determine the nature of that wood. And then we will also try to understand periderm. See, vascular cambium, there are two types of cambium that we will study. One will increase the thickness in the vascular region, whereas one will increase the thickness in the outer region, which will form the periderm, which is also a secondary tissue. So that also we will understand, and we will also try to understand the factors which affect this cambium activity, because it's a meristematic tissue. Okay, it has activity, so there are different reasons which control the activity of cambium. So that all we will try to discuss. So first of all, in this first lecture, we will try to understand the vascular cambium. Okay, primary meristem, okay, that we all have studied, is responsible to increase the height of the plant. However, in monocots, we know that we have a lot of time we have studied also, and before that in several lectures I have also told you that monocots do not have secondary growth. Okay, you would have seen the stems are quite weak, like wheat and rice and grasses. Okay, so they are very weak stem, so they don't undergo secondary growth. Even in case of some dicot which are very small, herbaceous in nature, even not all, okay, but very few of them may not undergo secondary division, and they could be limited by only primary growth. So whatever structure is present is of primary in nature. But in most of the gymnosperms and woody dicots, see, gymnosperms also, although we have not discussed about it, but gymnosperms also you would have seen that pine trees are there, cycas, pinus, that all you would have studied, and very large trees you would have seen in hilly areas. They all have very big stem. Okay, they all have very broad stem, very rigid stem. Similarly, many dicot plants, means all woody dicot plants, they all have very big diameter of their stem and root, and this is so because the plant is very big. So definitely to support them and to help them in conduction of water, minerals, and the food materials to different parts of the plant, definitely they need more elaborate vascular tissue. So because of that, they develop amount of secondary structures, and this growth is known as secondary growth, and it is with the help of cambium, which is also known as lateral meristem. So that also we have discussed in previous lectures, that cambium is also known as lateral meristem because it is found on the lateral sides, okay, as compared to apical meristem, which we have found in the apical, in the tips of shoot and root. So lateral meristem is found on the lateral side. Okay. Now there are two types of cambium. We always have discussed about cambium which is present between xylem and phloem. That also we have discussed when we were discussing about vascular tissues, how vascular bundles are arranged. We would have discussed that there is an open nature of vascular bundle in which you have a cambium, okay, and you have you have phloem which is there on the outer side and xylem on the generally this is the case in stem, okay, in dicot stem as well as in monocot stem also, at least the arrangement of xylem and phloem is similar, but in monocot you will not find this cambium. So, in monocot you have closed type of vascular bundle, whereas in dicot you have open vascular bundle. So, this is the cambium which is found between them. Now, this cambium will give rise to secondary phloem and secondary xylem. But see, when tissues are increasing, suppose this is a cambium here which is increasing xylem on this side and phloem on this side. So, the tissues are increasing. So, when the tissues will increase in diameter, they will push the cells which are present on the outer side. Now, with this increase in mass, definitely if the outer cells are also not increasing, then they will be completely broken down, they will be completely crushed. So, to accommodate this tissues which are increasing from the inner side, the outer layers will also increase in diameter. Now, this increase in diameter is brought about by another cambium which is known as cork cambium. Okay. So, this cork cambium originates from the outer layer of the cortex, okay, generally from the outer layer, and then it will increase the diameter from the outer side. Now, you can think of that what are the let us just try to revise what are the primary tissues and what is the primary meristem and what are the lateral meristem. Now, we all know about apical meristem, okay, they were there on the stem and root. Now, apical meristem was actually divided into 3 meristem. They were all primary protoderm. So, that all we have talked about several times. Protoderm, then you have procambium, then ground meristem. Okay. So, these are all primary meristem. Now, they will give rise to primary tissues. Protoderm was responsible to give rise to epidermis. Okay. Procambium was giving rise to xylem. You can, in the order you can write down. They were giving rise to primary phloem, vascular cambium and primary xylem. So, they are also primary tissues, okay. This primary meristem give rise to primary tissues. So, actually you can see here that vascular cambium is the primary tissue, means primary tissues, those tissues which are found from the embryonic stages itself, like from the early developmental stages, these tissues are present and they will be present till the end of that plant. Now, this vascular tissue will now behave as lateral meristem. Now, ground meristem, similarly ground meristem is also forms cortex and pith, okay. These are all primary tissues. Now, in the cortex another cambium which is known as cork cambium, various other names are also there, so that we will discuss when we will talk about it. Cork cambium. Now, vascular cambium will give rise to secondary tissues, that is secondary phloem and secondary xylem. And cork cambium will give rise to periderm, okay. Different layers are there. So, overall we call them as periderm. So, these are the secondary tissues and this is the secondary meristem. Okay, so in this way different layers of cambium will be formed. So, now in this lecture we will try to focus only on vascular cambium. Now, vascular cambium which is there between xylem and phloem, okay, This is an unspecialized meristem. It is a meristem, it is a layer of tissues which are not specialized, and they will give rise to specialized tissue, xylem and phloem. And this is a single layer of meristematic tissue. See, you should remember that cambium is a single layer of cell. Okay, suppose we draw the cross section, then only a single layer of cambium you will see. Now, if damaged, it cannot be rejuvenated, because cambium, because see, cambium is giving rise to other cells, but there are nothing else which could give rise to cambium. So, it has to be protected. However, there are various ways also, when sometimes if some layers of tissues are damaged, not cambium, but there also some sort of meristematic growth formation can occur, but as such cambium, if it is damaged, it is difficult to form again. Now, activity of vascular cambium varies. See, in some plants it will be functional for a short period of time, but in many large woody dicot plants it is functional for several years, even like decades or even in centuries. You would have observed some trees are have a very, they are so aged, means they have a very long lifespan, and so in these trees also you can see that cambium is functional and it is still giving rise to secondary tissues, and still you can see that the stem diameter is increasing, and you would have observed some trees with very huge diameters. Okay, so in that case definitely the cambium is active for very long time. Now, in plants which do not have secondary growth, so definitely what happens to procambium? See, procambium was giving rise to cambium, okay, and the primary phloem and the primary xylem, but in those tissues like in monocot, if there is no further secondary growth, so what happens? This cambium completely utilizes itself and forms primary phloem and xylem. Phloem and xylem. So, here you can see that there is no cambium formation. So, all the procambium layers have been utilized, and if there is no cambium, so definitely no secondary growth. But in those plants in which secondary growth formation is there, some part of the procambium is still present in the meristematic form, and it remains meristematic and give rise to the cambium proper, that is the vascular cambium. Okay. Cambium, by division, it produces the secondary vascular tissues. Now, how this vascular cambium was developed? See, as we all know that it develops from procambium. Suppose this is the shoot apical meristem. This is the shoot tip, and here you will have the apical meristem. We know that apical meristem will give rise to layers of tissues, okay. So, outer one will be the epidermis, okay, or will be known as protoderm, okay. Then you have cambium, then you have ground meristem, okay. So, this procambium actually, this procambium is found from promeristem, that also we have discussed in our previous lectures. Promeristem. See, promeristem is basically the embryonic meristem, means it is found in the embryo. From the embryo, when it starts developing, there is a region which first of all it gives rise to promeristem, it is completely unspecialized. Then this promeristem will give rise to primary meristem, that is all the apical meristem, all the layers, okay. Now, what happens? Suppose in the initial stage primary promeristem is there. So, what happens? Now, in promeristem, cell divisions occur. Cell divisions occur without actually increasing the diameter, okay. So, thin cells are formed. This, let us just elaborate it. In the promeristem, lot of periclinal division will occur. Okay, periclinal, as we have discussed before, which is increasing the diameter. Okay, suppose any division which is increasing the cells on the outer layers or in the inner layers, so that is periclinal, and anticlinal division is one which is just increasing the surface layer. So periclinal division will occur. Okay, so some cells will be differentiated into ground tissue. Then you have a protoderm. Okay, see, this is just, you can say it is an, so outer layers will be protoderm and these layer will be procambium. So in this way, promeristem will be differentiated. Now what happens, see, when we say procambium, see, there are several transition states also. Okay, when procambium is formed, it's not that it is just abruptly giving rise to, see, there could be different transition state, and different names could also be there. Sometimes we call it as provascular tissue, provascular or residual meristem. So what happens when this procambium, suppose it is forming a layer, in procambium, in that also there are some densely stained region. Okay, when scientists were observing it, they found that some densely stained regions was there in that residual meristem or that intermediate stage. So they called it as fascicular cambium. Okay, some densely stained region between that residual meristem or that provascular cambium layers, or it is also known as intrafascicular cambium. See, this is all very important when we will study the secondary growth in stem, and those regions which were between those densely stained regions were known as interfascicular parenchyma. So this fascicular cambium is actually the cambium which is found between, so we call this cambium which is found between the xylem and phloem as fascicular cambium. Okay, and the cambium which is between, see, actually you would be thinking that actually there is no cambium in between the vascular bundles. Okay, vascular bundles are arranged as such in dicot. You will see that they are arranged in the form of ring. So as such there is no cambium, but when secondary growth formation will take place, then the layers between this, the layer which I was talking about, interfascicular parenchyma, will give rise to interfascicular cambium. Okay, so then a complete ring of cambium will be formed. So that means the fascicular cambium is actually primary in nature, which will originate, which will be present from the time when the cambium formation, procambium formation took place, and interfascicular, it will be actually secondary in nature because it is arriving at later stage of plant growth. It is not present from the beginning. Now coming to the structure of cambium. Now see, vascular cambium, we will talk about vascular cambium. Okay, we will talk about this vascular cambium. See, it is single layer of meristematic tissue made up of two types of cell. Suppose this is the stem. Okay, so inside also it will have cambium, which will also form a cylinder like this. Now try to, this is just a single layer of cylinder. You just imagine a single layer of sheet which you can fold it up in the form of a cylinder. Okay, so this is all single layer. So just try to understand in the longitudinal. See, this is the longitudinal section and this is the transverse section. Okay, so try to imagine this cambium layer in the longitudinal section. So what you will observe, you will see that two types of cells are present in the vascular cambium. One type of cell is elongated and have tapering ends. Okay, so these type of cells are known as fusiform initials Okay, you can say that they are tapered and they are vertically aligned in the axis to vertically aligned towards the vertical axis. See, this is the axis, okay, vertical axis. So, you can say that they are aligned vertically on the axis, okay, and these fusiform initials will produce all the layers of all the secondary layers of xylem and tissue, means all the secondary tissues of xylem and phloem will be produced by this fusiform initials. So, xylem, definitely what are the components of xylem? You have vessels, you have tracheids, then you have fibers, and then you have xylem parenchyma. Okay. So, this fusiform initials will give rise to them. Then phloem also, you have sieve tubes, you have companion cells, then you have fibers also, and you have phloem parenchyma. Now, these secondary tissues, so it will produce secondary xylem and secondary phloem. Now, these secondary tissues will be utilized in the vertical transport, okay. In the vertical flow of all the materials, like xylem will transport water and minerals. Similarly, phloem will translocate the food which is prepared from the leaf. So, it will be used up in the vertical transport. Now, coming to ray initials, now in this layer, okay, this single meristematic sheet like structure, you will also observe some very small cells, okay, very small cells. They are known as ray initials. Okay, they are shorter cells, generally isodimetric, means their diameter is almost similar from all the sides, and they will generate cells of the radial vascular system. Now, earlier also I have told you that there are two transportation systems, one is towards the axis and one is radially on the radial side. So, this is the radial transport and this is the axial transport. So, axial transport, we have talked about that all the components of the xylem and phloem, they will all take part in the axial transport by fusiform initials, they will be generated. Then in radial transport, ray initials will be generated. They will be these ray cells, okay, they will also be produced from the cambium, okay, through pericrinal division, but they will be oriented in the radial pattern and they will transport substances in the radial direction. See, they are made up of parenchyma cells because they are thin walled, and parenchymatous cells are always thin walled, they are not highly lignified. Generally, the purpose is sideways or the radial transport, and since they are also parenchymatous, they can also perform function in storage. Now, they are known as xylem rays when these rays, when these rays are present in the, these rays, you have a ring of xylem, you have a ring of phloem, okay. So, these rays will also be present in the xylem. So, since they are radial, so they will traverse the xylem, they will also traverse the phloem. So, when they are originating in the xylem, they are known as xylem rays. When they are forming in the phloem, they are known as phloem rays, okay. So, definitely in the xylem and phloem, the parenchyma tissues, which we know that they also have parenchyma. So, basically they have two types of parenchyma. Parenchyma. One will perform the axial transport and other will perform the radial transport, okay. So, they are also component of xylem and phloem. Now, let us try to understand how these fusiform and ray initials are arranged in that cambia. See, on this two basic patterns are there on the basis of fusiform initials. See, initials we call because these are the cells, initial cells which are giving rise to secondary xylem and phloem, okay. Now, storied cambium, in this you can see here that the ray initials are parallel to each other and they are aligned on the same plane. As you can see in this case, they are all aligned in almost in the same plane, Then in non-storied cambium, you can see that they are not aligned in the same plane, okay, like this, somewhat coming like this. So, they are not aligned in the same plane. So, this is storied and non-storied. Similarly, for ray initials, like these small rays are there, okay. So, you can say that they could be uniseriate if they are arranged in single files. They could be biseriate if two files of there, and they could be multiseriate if you can see multiple layers of vertical layers of ray initials are there. So, they could be this different type of orientation, as you can see, uniseriate, then biseriate, okay. Then how these fusiform initials are formed? See, for fusiform initial, simple anticlinal division, okay, simple anticlinal division will give rise to two fusiform, okay. So, it is very simple that they will give rise to two, okay. Similarly, a single fusiform initial, okay, they can be divided through oblique septa. So, in this way, so this will give you storied arrangement. For non-storied, there will be an oblique division, and this division can be intrusive, and then it can give rise to non-storied. So, you can say that oblique division, and this division is extending towards each other. So, this will give rise to initials which are not on the same orientation. Then you have then ray initials, how they are formed. A single fusiform initial, if it is divided into different, it is by transverse division, it can give rise to several ray initials. Similarly, single fusiform initial can divide with oblique septa, and there also multiple division can give rise to ray initials. A single fusiform initial can be reduced to ray initial. So, this is the way how they are formed. Now, in the last we will describe about what are the cell divisions which is taking place in vascular cambium. So, two types of divisions are there, periclinal as well as anticlinal. See, in periclinal, as I have told you, suppose this is the cambium. So, it will cut tissue in periclinal way, increasing the diameter, okay. Suppose one cambium is there, okay, it is dividing into two cells, one will remain as cambium, one can form phloem. Then again, this will again divide to form xylem, okay, again this will remain as cambium and this will be phloem. So, in this way several tissues will be cut on either side, with it will increase the diameter. Now, in the beginning, see, actually cambia is single layer, okay, it is single layer. But in the beginning what happens, the division is fast and the initial phloem and xylem cells which are formed are not differentiated well. So, you can see a cambium zone. So, this is known as cambial initial because or a cambium zone. So, that means a cambium zone you can see of undifferentiated cells, they could be also known as phloem mother cell or xylem mother cell. So, this cambium initial cell, so it will look like multicellular, although the cambium is actually single cell, but a cambium zone is multicellular. Now, another function is through anticlinal division. So, what happens? So, suppose this is the cambium cells. So, it will also divide anticlinally to increase its number. See, when cambium is producing xylem and phloem, so to accommodate the tissue, see, xylem is accommodating inside. So, to accommodate the tissues, it has to increase its diameter also. So, they will divide anticlinally to give rise to increase the surface area, to increase the diameter of the cambium. So, in this way cambium will divide to give rise to secondary tissue as well as to increase the number of its own cambium. So, now this figure just only shows that how this fusiform initial is divided through periclinal division. Periclinal division you can see here, and it is giving rise to all the xylem, sieve tubes, vessel, tracheids and all. And see, this is the ray initial, this is also dividing by periclinal division, but it is giving rise to radially arranged parenchymatous cells. So, this is all for today. Thank you. We will meet in the next class.